Good afternoon, everyone, and welcome to this special webinar on advancing clinical interpretation from single-cell datasets. I'm really excited about this. My name's Diana, and it's a pleasure to be introducing you to today's speakers. But before I do, I have a couple of very quick announcements to make. First, I want to draw your attention to the questions tab on the right. This is the best way to ask questions during the webinar, and we have got a live Q&A at the end. You can also upvote the questions from other people that you like, and we'll have plenty of time, so please do put them in as soon as you think of one. We also will be sharing this webinar on demand, but we will not be sharing the slides from today. So you can access the recording any minute from today's ending and just use the same link that you used to come on today. So I'm just going to hand it to the speakers for kicking us off. Ju, would you like to introduce yourself and the work that you do?
Definitely. Thank you, Diana. So my name is Ju Fan. My background is in bioinformatics, and I have been working in the single-cell field for more than 8 years. So currently I'm working for Singleron Biotechnologies. I'm responsible for bioinformatics and data science department. And we are actively developing products to help people analyze and interpret their single-cell data. I'm very excited for the webinar today, and also very excited in learning tools developed by Antonio and his colleagues, and these tools' clinical applications. Yeah, thank you.
Thank you, Ju. Antonio, would you like to introduce yourself and kick us off with the presentations?
Sure. So this is Antonio Rose. I'm group leader of the Clinical Bioinformatics Laboratory at the Imagine Institute for Rare Diseases, located in Paris, France, and I will be presenting today some of the new computational approaches that we have been recently developed for the unbiased identification of molecular signatures from single-cell sequencing. I will be presenting together with a postdoc in my lab, Marceau Quadre-Dernier, that will present later on as a part of the talk. So first of all, I would like to disclosure, both for Marceau and myself, that we have no actual potential conflict of interest, and we have no relevant financial relationship or commercial interest to disclose. So in terms of the outline, I will be just motivating my talk, then I will present CellID, which is a recent tool that we developed for the per cell molecular signature extraction from single-cell RNA-seq data, then how we applied this approach for the characterization of human kidney cell heterogeneity, and finally I will present novel developments such as the approach sample ID for the sample-level molecular signature extraction. So moving forward, as a small introduction to the audience, there's a number of large-scale single-cell RNA-seq projects that are being developed by a large consortium and international initiatives such as the Human Cell Atlas, the HubMap project, or also in model organisms such as the tabula moris. This mostly is devoted to healthy samples, but there's also initiatives that are mostly focused on pathogenic samples covering a number of diseases such as lifetime initiative. So just to cite some examples of the discoveries that were obtained thanks to single-cell approaches is a more complete atlas of human blood monocytes and dendritic cells, as well as microglia subtypes associated with Alzheimer's disease, that were recently, initially described thanks to single-cell approaches. My laboratory has been involved in the development of computational tools for single-cell analysis. So we published a number of years ago software called SynCell to provide statistical significance for cell trajectories from single-cell RNA sequencing. And then applying these approaches, we identified subsets of T cells that are highly enriched for susceptibility to HIV infection. This was one of the first approaches of single-cell sequencing applied to the identification of cells related to susceptibility to viral infection. So just to give in a nutshell a brief introduction of the computational approach to identify relevant disease-associated cells. So typically you start from a number of single-cell sequencing experiments from different samples, and at some point you may want to perform a comparative analysis between healthy and pathogenic tissues. And for this, you typically need to do some steps of batch effect correction, dataset integration, dimensionality reduction, and clustering in order to identify specific cells that can be cell types or cell state that are associated to pathogenic samples while they are absent from healthy tissues. So once you've identified this subset of cells, you can do differential expression analysis in order to identify potential biomarkers or molecular signatures that can help understanding which are the functional specificities of such cells. However, these approaches have a number of challenges regarding how data is integrated, how batch effects are corrected. Clustering is a tricky computational step. And then we wonder whether we could propose a computational approach that would be able to assess each sample and each cell on its own, just overcoming the pitfalls of the integration and the clustering steps. So for this, we first developed an approach that is called cell ID, that is able to extract signatures and then predict cell identity recognition for each individual cell in a dataset. This was published some months ago at Nature Biotechnology, and I will briefly explain the approach. So basically, when you start from a count expression matrix, where you have, on the one hand, the gene expression levels for each gene and then for each individual cell, you can perform through an approach that is similar to principal component analysis, that is called multiple correspondence analysis, a low-dimensional projection of cells, the cells in your specimen, which are represented in the figure by orange circles. However, with as a peculiarity of MCA, you can simultaneously project genes together with cells in a way that the closer a gene is to a given cell in that low-dimensional space, the more specific to that cell the gene is. This allows, in turn, to assess distance for each gene to each cell in your sample, so that you can rank genes for each individual cell in order to obtain the most closely associated genes for each cells that are represented here by a yellow diamond that we will define as the signature defining such cell, some sort of a molecular identity cover. So this can be done for each cell in a sample, but in parallel can be done for multiple samples independently. This is interesting because then this allows us to extract signatures for cells and then compare whether these signatures match with independent cells from other samples, allowing us to do cell-to-cell matching across independent datasets. But then we can also assess enrichment for specific cell type signatures using predefined list of cell type markers, and as well as functional enrichment analysis against reference marker lists from functional databases such as Gene Ontology, Reactome, so on and so forth. So by doing so in the article, we prove that cell ID is able to provide multi-class cell type labeling at individual cell resolution using pre-established signatures, which we illustrated here through the analysis of human cord blood mononuclear cells that were profiled using site-seq analysis, so that we can have some sort of a golden truth, allowing us to do a proper benchmark. By doing so, we projected this cell identity to each individual cell without, again, any clustering step, and then indeed obtain a very good matching with the purportedly bona fide cell types. So the interesting thing is that, for example, if we focus now in the subset of immature CD34 positive cells, we do see that indeed we can profile very well all the hematopoietic stem cell signatures transitioning from the most immature states such as HSC towards more mature states like erythrocytes. So as it is reflected in the figure, each individual cell can actually be enriched for more than one cell type at a time, showing this smooth transition. and this would be lost if a clustering step, for example, or a differential expression analysis is applied. So in the article, we show as well that CellID is able to extract signatures from very rare cell types, as for example, brass staff ionocytes, globed cells from airway epithelium, so that we can match the equivalent cells either in airway epithelium from other animals or from other species such as in human. As well, we can match these rare cell types from airway towards epithelium, intestinal epithelium, and we match the corresponding endocrine, goblet, and brass staff cells, as well as for the olfactory epithelium cells. This was very interesting to show indeed that in previously developed olfactory epithelium data sets in which solitary chemosensory cells had gone unperceived, by using brass staff cells of airway epithelium, we identified such SCCs that had been previously reported through bulk RNA-seq, but were not being reported previously at the single cell level. So by using single cell ID samples from brass staff, we identified a number of 37 and 5 SCCs from 2 independent olfactory epithelium datasets, and then by performing cell ID pathway enrichment analysis and individual cell level, indeed show that these cells are indeed enriched in cysteine leukotriene biosynthesis pathway, as well as in IL-17 signaling pathway, which are characteristic of the SCC population. So in the article, we saw as well that CellID is able to work at a large scale by handling multiple organs and cell types at a time, and doing so as well in a robust manner when matching cells across different single cell technologies, as represented in the figure, matching single cell RNA-seq and single cell ATAC-seq data in mouse. So now we are show more recent work in which we applied the CellID approach to the study of renal ciliopathies. This is in the context of a project where we are interested in characterize the heterogeneity observed at the clinical level across samples from these renal ciliopathies, by which some of them will develop end-stage renal failure early in life, while others will do it much later. So the interest is to first describe this heterogeneity at the cellular level in order to identify the proper window of therapeutic opportunity. This is going to be presented by Marceau, so I let him the floor for this part.
I move my mask. Thank you, Antonio. Hello, everyone. So, yes, as Antonio said, to introduce, while working in the renal field, we identified that there's a lack of reliability between the studies, mainly because the cell types are not attributed to cells the same way, depending the studies, in both single cell and single nucleus RNA sequencing datasets. So we aim to design signatures, consensus signatures of renal cell types, and use it in every coming studies, or can be used in every coming studies in the field to increase the reliability. And so we followed, and to do that, this workflow where we collected the data for both single cell and single nucleus data sets with a total of 32 and 7 samples respectively. We computed, after processing of the data, we computed consensus signatures in both conditions, and I will show you the test of these signatures on previously published data sets. So to do so, we aggregated available samples from the literature, and for both single cell and single nucleus data sets with a lot of cells and quality nuclei from both 32 samples for single cells and 7 samples for single nucleus data sets. We clustered these data sets with the classical algorithm and attributed the cell types based on the known marker from the literature in those data sets. So here you can see single cell on the left and single nucleus on the right with the annotation of cell types we found. We we then computed the signatures of single cells datasets with a total of 28 consensus signatures. And for single nucleus, it was a total of 22 signatures. And then we just integrated single cell and single nuclei datasets to see if the cell type matched between the two experiments. And as you can see here, the annotation of both datasets are consistent. So the signatures we got from both single cell and single nucleus seems to converge. So now we know that we have good signatures. We can apply it on previously published datasets. First, here's the single cell dataset published by Christoph Cooper and colleagues. And on the left, you can see the annotations of the authors, the original annotations. And on the right, the signatures we enriched using cell ID. And as you can see, the correspondence is very high. The main difference is that some endothelial cells from the original annotation are now labeled as B cells, depending on the signature we have. For single nucleus datasets, we applied also the signature. We enriched the signatures, and as you can see, the correspondence is even higher with very good annotation from cell ID using our signatures. Some cells are not attributed, but come from the same individual, so cell ID may even perform better than what we could see before. Thank you very much.
Thanks, Marzo. So far, we've been talking about the extraction of molecular signatures at the individual cell level with cell ID. An additional question we had in the laboratory is whether we could extract unbiased molecular signatures, not at the individual cell level, but this time at the sample level. And this is in order to classify or to stratify patients according to the general profile observed across the individual cells. So the goal is then to extract a fingerprint of the genetic association with the cell heterogeneity observed within a sample. And how we did do that? So basically, we start from a gene expression matrix where we again have gene counts for each individual cells. And then we can, in a way, partition the total heterogeneity into independent factors. Now, we can, for each independent factor, evaluate whether the information explained is actually different from background knowledge, allowing us just to retain the amount of information that is relevant to explain such heterogeneity. Once we have done that, we can partition each fraction of the variability explained by each independent factor and attribute it to each individual gene detected in the analysis, in a way that for each independent factor, we assign the fraction explained by each gene. In addition, we can then add at the gene level, which is the fraction of the total initial variance that then is explained for a given gene, that's represented in the figure. By adding the different contributions to the total variance, then we can assess what we call the phi score, that is somehow the importance of the gene to explain the cell heterogeneity in a sample. We can then rank genes according to their phi score, and then, as mimicking what we did for cell ID, we can obtain for the top ranked genes signatures that this time is at the sample level. These signatures are useful because then can be used as in cell ID for an automatic annotation, functional enrichment analysis, and then matching samples against a reference database of single cell libraries. So going more into the mathematical details, the approach that we built is based on singular value decomposition so that we can get orthogonal factors and then distribute and weight for each gene the variance explained according to the total variance explained in each independent factor. So this is unpublished data, and we are still working on the characterization of how the methods behaves. For this, we first started by analyzing single cell libraries from the Tabula Muris project for which there are multiple samples available for a number of organs, and interestingly, there are several samples for each represented for each organ represented. So by applying sample ID, this allows us to extract these signatures at the sample level and then see how these gene signatures correlated across samples in a way that, as shown in the figure, samples cluster according to the tissue of origin, which is a first reassuring result, showing the ability of sample ID to get signatures that are reproducible across independent samples from the same tissue. We then have applied the sample ID approach to the characterization of single-cell RNA-seq samples from autoimmune disease patients, where the goal is to try to stratify or classify patients according to their clinical status. I cannot give more details about the specific disease and genes involved, but the figure shows two families affected by this autoimmune disease caused by the different genes on the left than on the as compared to the right family, and in both cases with incomplete penetrance, meaning that you can find members of the family that, while bearing the causal mutation, they did not develop the disease. So we perform a single-cell sequencing analysis from blood cell samples of the individuals highlighted with colors, and then see how these samples cluster according to the sample ID signature that was extracted in an unbiased manner. Interestingly, for some individuals that are highlighted in green, these were patients that had been treated so that a remission to a healthy status had been observed. And this is interesting because for some patients, then we had both samples before and after treatment, and then we were able to see whether their sample ID signatures had actually become close to normal. So these are the results that we obtained by applying a low-dimensional representation of the sample ID signature of the theme for each of these samples. So as it can be seen, all healthy carriers, or actually non-carriers, clustered together on one side of the low-dimensional representation, as well as those samples from patients having been treated where a remission to a healthy state had been observed. On the contrary, samples from the patient before treatment from one of the families and for the other patient that had not been treated mapped far from the central cluster. So again, these are reassuring results. We are further working on the characterization of the cell ID approach and the clinical value for matching samples against reference libraries of patients. This is a topic that I think that will be mostly approached in the next presentation. So just to recapitulate as take-home messages, just to say that cell ID and sample ID allow automatic single-cell data annotation using reference databases. This allows us also to do an unbiased gene signature extraction at cell and sample level. It has been highlighted during the presentation for human healthy kidney samples. Also that both approaches allow a multi-experiment cross-mapping, overcoming the pitfalls of batch effect correction and data integration, because here in both cases we applied approaches for each individual dataset independently. Then that the cell ID approach allows ultra-rare cells identification, and that both approaches allow automatic functional annotation enabling discovery, meaning that when you don't have a good reference gene marker list in order to identify a given cell type or cell state, you can still do this unsupervised approach, so that once you have identified specific cells or specific cell subpopulations, you can extract these gene signatures, and then by applying functional enrichment analysis, characterize whether they are, for example, in an inflammatory state or in a apoptotic state, so on and so forth. So this CellID software is open source. It's available at the GitHub repository of the laboratory, and Sample ID will be soon released as well as an open source software. So you can stay tuned at the Twitter account of the laboratory. So I thank you for your attention. I would like just to acknowledge the work of Akira Cortal and Marceau Dernier, on which it was based most of my presentation, as well as the work from Loredana Martinetti regarding the sample ID part. Thank as well all of the members of the laboratory for interesting input and feedback, and collaborators at the Imagine Institute. Acknowledging as well the different grant funding sources, both public and private, of the lab. Thank you all for the attention, and I will be happy to ask questions.
Thank you very much, Antonio, for that very interesting presentation. Thank you for the questions as well. Please do keep them coming. I'm now going to hand the virtual stage over to Ju for our second presentation. Ju, over to you.
Okay, thank you, Diana. It's actually my pleasure today to have the opportunity to share our recently launched product, single-cell database, Synaposis. And today I will also talk about the public single-cell data mining strategies empowered by Synaposis. So as people know, single-cell sequencing has been a hot topic last decade since its debut in 2009, and especially since the commercial products launched in 2017. There are already thousands of publications published in the scientific community. And more recently, in the last few years, the technology has moved more into the translational and clinical research. Actually, if we search on clinical trial website, we can see there are already more than a dozen clinical trials sponsored by world's leading pharmaceutical companies utilizing single-cell sequencing technology. And from the box sequencing era, we know that mining the huge collection of patients' sequencing data has resulted in great improvements in the precision medicine. For example, people now can couple the driver mutations in lung cancer with targeted therapy for its treatment choice. And also, for example, in breast cancer, people can use the expression profile of patients to divide them into subtypes and treat them accordingly. And in this more precise era of immunotherapy and cell therapy, actually single-cell sequencing can give you a more precise cell type level gene signature of patients, and we can associate both gene level and cell level signatures to patient subgroups. So the translational researchers and also R&D scientists, drug R&D scientists are interested in mining these already existing thousands of public single-cell data in the public domain. But currently, there are a lot of barriers preventing people to utilize these huge data sets. For example, different publications sometimes use different platforms and analysis pipelines to generate those data, and they label their cells differently, which makes comparison between data sets very difficult. And another barrier to prevent people mining those data is single-cell sequencing currently still require a relatively high level of bioinformatics skills to analyze them. So to solve those problems, we Singleron Biotechnologies newly launched a database called Synaposis to standardize those single cell data sets. And we also provide easy to use data visualization tools for people. We hope people can, without bioinformatics skills, can use this database to mine their interested information more easily. And this is the landing page of synaposis.com, and later in this presentation, I will give you a little tool about the website to show you the functions and the interface of this database. And we specifically designed the database to solve the current main pain point of the current available single-cell database. So the current open source and commercial database usually only collect the sequencing data itself without much metadata associated with each sample. And without those clinical and molecular level metadata, it's actually very hard to use them for translational or clinical purposes. And to solve this problem, we actually try to curate those information from the public domain as much as possible. You can see in this example, we collect the molecular level information such as gene mutations and their mutation site details. And for patients' treatment history, we have the drug information and their treatment responses. And for the diagnostic information, we have the their disease subtypes, their stages, and also for metastatic patient, we have their metastatic sites. With all these clinical and molecular level metadata available, that will enable the association between gene level and cell level information to patient subgroups. And another main pain point of the current single cell data analysis, which is also touched in Antonio's presentation, is that people usually only analyze a single data set at a time. A single data set usually refers to all the samples in one publication, and different publications use very different cell type annotation terminologies and also resolutions, which makes them very hard to compare to each other. So in our workflow, we try to use a very standardized and a workflow both for the analysis part and also the metadata collection. And whenever possible, we try to use the recognized standards for those metadata and also analysis parameters. For example, we collect patients' disease and disease subtypes according to disease ontology. And for the cell type annotation part, we use a uniform set of resolutions, and also the terminology are linked to cell ontology, which makes their definition very clear. Here is an example comparing the cell type annotation in the original publication and also our annotation in the Synaposis database. So you can see on the left is the author's original annotation. You can see they only annotate those to the immune major immune cell types such as T cells and dendritic cells. And they also lumped macrophage and monocyte into one cell type without further dividing them into subtypes. But in our database, we use the uniform set of the cell type and subtypes for all the data sets we collected in the database. And we would divide T cells, dendritic cells, macrophages to their corresponding subtypes. So we also offer functionality to let the customers upload their own data and use our uniformed set of analysis and annotation pipeline to analyze their own sequencing data. It ensures the comparison between your own sequencing data with all the data sets we collected, already collected in the database. So all the parameters and the annotation standards are validated by thousands of single-cell service projects we have been done at Singleron Biotechnologies. Actually, in this pipeline, we use Antonio's tool, Cell ID, for the auto annotation tool. In our benchmark, Cell ID is definitely one of the best auto annotation tools currently available. So next, I will go to Synaposis.com and show you a little bit about its interface and functionalities. And I will give you a few examples to see how you can use this tool to validate your scientific discoveries, to validate or discover for novel drug targets, and it's to find patient indication for your potential target. So let me switch to the website. So this is, if you go to Synaposis.com, and this is the landing page. And I already logged in my account. And if you are interested in learning more about the database, you can go to here and sign up a demo account of our database. So let's get started. You can see on the homepage of Synaposis, it shows some of the data statistics, how many data sets, cell types, marker genes, cells, and tissues we collected in the current database. So the main function is under this browse by section. There are 4 different functionalities. You can search based on tissues and genes, cells, and individual single cell studies. So let's go to Tissue Atlas first. And we have currently both human and mouse data sets. And we divided the human body into 12 different systems. And under each system, you can look at the data sets, cell types, and markers associated with this particular system. And if you click on the cell type, you can see the major cell types, the cell subtypes associated with this specific tissue. And if we click on one subtype, you can look at the canonical markers of this particular cell type. And these markers are based on the knowledge base accumulated by Singleron Biotechnologies since it's founded and has been validated by thousands of projects we have been done for our customers. And if you click on the dataset part, you can directly go to the dataset search section. And if you're interested, you can go to view the individual single cell datasets. So let's go to the gene search part first. And now you can search for your interested genes and the single cell datasets associated with it. Let's say we try to search the drug target people are very interested in, for example, TIGIT. We can see on the first section, it shows some of the basic information of this gene and the pathways it's associated with, and the drug target in a public database. And the next section is a database-wise distribution bar plot of this specific gene. So because we standardize all the data sets, we can compare the data between hundreds of data sets in Synaposis database. So based on all the data sets in our database, we can see the tissue-resident memory T cells has the most expressed genes percentage for TIGIT, and actually very consistent with the current knowledge. We can see multiple other T cell subtypes have very high expression of TIGIT. And if you want to see what kind of disease associated with tissue resident memory T cells expressing TIGIT, you can click here and you can look at the different disease associated with the tissue resident T cells expressing this particular gene. You can see there are different cancer subtypes and also non-cancer disease. And we also offer the functionality to search for cells. So if you want to have a cell-centric view, you can click on the cell type search here. Let's say we search for Tregs. And also we have this section like briefly describe this cell type, and the next section show you the canonical markers that we can use for both human and mouse to identify Tregs. And similarly, we have a database-wise distribution to summarize all the information based on hundreds of data sets in the database. We can see like in the different tissues, Tregs have the different levels of infiltration, and we can also look at the disease associated with this specific tissue if it has Treg infiltrating in this tissue. And if you're interested in the individual single cell data sets, you can click on the study search to search for both the paper in publication information, and also you can filter for different kinds of samples, like different species and tissues, and you can also filter with those clinical metadata fields. And for each data set, you can click into the view to have to look into details of these data sets. And there is a section called Synaposis core data sets here that we pre-selected high-quality data sets associated with a certain disease and pre-compiled them into 1 mega data sets. For example, we can click into this lung cancer data sets. You can see this already pre-integrated core data sets has more than 300,000 cells. It has 170 different clinical samples, and it was based on 4 different publications. And for each publication, if you click into the view section, you can look at the paper information of this specific data sets. And if you go to analysis page, you can look at the UMAP plots of this data set. And you can also look into the cell composition, the gene expression details of each gene in this data set. And very importantly, you can use the differential expression functionality to compare different cell subgroups. For example, you can use this function to de novoli find potential drug targets for, let's say, because this data set contains the lung cancer samples treated with targeted therapy. If we compare the responders with non-responders for the different cell types, we may find the combined therapeutic options for the targeted for patient treated with targeted therapy. And also, like I introduced previously, we have a data analysis portal that can let the customers update their own gene expression files and then use our standardized analysis pipeline to analyze and annotate their data sets. So here we actually use cell ID as our default option for the auto annotation tool, and we also use other tools as options. And we use cell ID for the algorithm, and we use the Singleron curated reference for different tissue types. And after people choose their analysis parameters, they can submit their analysis. So let's go to one of my analysis I submitted a few days ago. You can click the view results part, and you can compare the cell type suggested by your algorithm, associate the clusters with your own analysis, and you can actually adjust or custom the analysis to have the final result of your cell type annotation. And the visualization part, the interface is very similar to the interface of the public data sets. So based on those standardized analysis pipeline and annotation pipeline, this will ensure a very reproducible and results for your single cell sequencing data. And it's very easy to compare your data with all the collected public data sets in the database. This is all my presentation today. And if you are interested in try out this database, you can directly go to synaposis.com or contact us through our website. Thank you.
Thank you very much, Chi, for that very interesting presentation. We have a lot of questions to go through, so I will get started on those, but I just want to invite the audience, if you have any last-minute questions, please do put them in the questions tab, as we'll be working through those. So, Antonio, one of the questions that came up for you was, what is the advantage of cell ID compared with other rank-based gene signature methods?
So the main advantage is that cell ID is able to work at individual cell level. So assuming that you even have one cell of a given rare cell type or a rare cell state, you can still analyze it, extract a signature that maybe you don't know which cell type it is, but still you can characterize it against functional terms, functional pathways, so on and so forth. So basically, it provides the highest resolution that is available, and let's say it allows functional characterization even when the corresponding cell type is not allowed. In the paper, we did some benchmark against other methods that can still do some part of the job. They can eventually annotate individual cells, but not necessarily perform functional enrichment analysis, or the other way around, or they can use pre-established cell signatures, but not necessarily cell matching, etc. So we did a complete benchmark for each of the scenarios against the state-of-the-art methods, and in most of the cases, CellID was outperforming available methods. So I invite you to go there and see in detail the results.
Thank you, Antonio. Could you also explain a bit about your clusterization approach?
So I think that the cluster part corresponds to the human kidney samples, right? So basically this was based using standard approaches like available and in the Surat pipelines using KNN and so on and so forth. So it's quite classical approaches in this case. Yeah.
Thank you very much. We had a question that came in about how many cells should optimally be sequenced to have a good view of differentially expressed genes in highly heterogeneous specimens, such as cancer. Would either of you like to make a comment on that?
I think this would be a better question for you, I guess.
Yeah, I think it depends on your scientific questions, because yeah, indeed, cancer is a very heterogeneous specimen, right? But there are a lot of cell types, they have very low percentage. If you want to, or you are very interested in those very rare cell type, maybe you have to first do an enrichment for your cells in the cancer and then study those cells. Otherwise, you can look at the subtype of those rare cell types. So I guess it depends on your the question you want to answer, and I mean what kind of experimental design you have.
Thank you very much. So this question had a lot of upvotes. So how did you perform your single cell meta-analysis? How is it different from integration?
Can you repeat, please?
How did you perform the single cell meta-analysis, and how is it different from integration?
Okay, so basically we use a multivariate analysis in a way that unspecific genes go to the origin of the space, while genes that are specific to a given cell type or cell state go close to those cells. So let's say that anything that relates to a batch effect or things that are not particularly associated to a given cell state or cell states won't be highly ranked at the per cell gene signature. So in such a way, these signatures are somehow robust to those batch effects, even if they exist, because the genes associated with them won't be part of them. And then this allows us to do this matching across samples, either at the sample level itself, such as in sample ID, or at the cell level with cell ID. And we show that indeed both methods are highly robust to different batch effects coming from the use of different technologies, model organisms, individuals, etc. So that's the way it works.
Thank you very much. Ju, I know you answered this in the chat, but I just wanted to bring it up for those who didn't see the answer. So it says cell ID can do cell annotations. How is it compared with single R, for example?
So Singular was actually included in the benchmark, and for most of the analysis we did, CellID is outperforming it. So we were reassured by that.
Thank you so much. So what database does CellID use to annotate cells?
So we don't preconize the use of any specific database, because depending on the tissue, organ, or even model organism you are looking at, you may want to look at specific reference databases. So it's difficult to provide general recommendations to be used by default. We don't think that's our job to develop such databases, so we just provide the tool, and then it depends on the specific question you have. The user can choose different possibilities. So nonetheless, I would say that good general purpose reference databases can be the human set atlas for healthy tissues, especially also the CART or the SCART reference database for single-cell RNA-seq but also ADAC-seq samples that have been developed at different embryonic stages, which is extremely interesting for the study of developmental disorders. And then in our studies, we have used reference databases such as XCell for immune cells or PanGLAO database for other tissues. But again, we don't have a special recommendation here, and I think the users will have most probably a better criteria depending on their program.
Thank you, Antonio. One of the questions that came in was, how do you identify ultra-rare cell types? And obviously needing a package for using those types of cells. Could either of you comment on that?
Sure. So basically, you can either use pre-established gene marker lists if those rare cell types are already identified. So you can use that as an input for cell ID. But you can also do discovery of rare cell types. So this means that you will identify through your analysis particular cells or few cells that do not behave as the rest, in terms that they are unassigned using reference marker list. You can then see which functions they are enriched for, and then whether, for example, they show signatures for specific pathways that can be meaningful depending on the specific disease you're looking at. For example, if you're in the context of cancer, you may want to look for immune infiltrating cells, or for specific apoptotic cells, or cytokine producing cells, so on and so forth. So basically, when you do discovery, you don't have such reference, but with CellID you can still characterize which genes define those cells and which are the functions that go associated. So it's the starting point to a discovery of a new rare cell type.
Thank you very much. This question I came in, I think it's for... Yep, go on, Ju.
Yeah, I just want to add to previous question and this question, these two previous questions, all about the reference database. I think actually references are very important and tricky part of all the auto annotation jobs. Actually, first you have to know a lot. You have a great knowledge about your tissue, like what kind of cells could be in this kind of tissue samples. And sometimes we find that one particular public data sets, they don't include some of the cell types normally in this particular tissue. So sometimes we combine multiple public data sets to have a more comprehensive set of cell type representatives for this particular tissue. Because if you don't include some of the cell types in your reference, you will only get like unassigned or very low prediction power of your cell type. So I think it does take a lot of work to build a reference database that can work very well.
Thank you, Ju. One of the questions that came in during your presentation was, which countries are your samples coming from, and to what level do you integrate this with clinical data, and what variables are you routinely annotating? Do you have anything to say on that?
So for Synaposis database, we actually use all the public data sets. So whatever the authors published in their country, we collect those data sets publicly available. It's just different publications usually sometimes hide those metadata in different places, sometimes in their attachment or sometimes in the data repository. Or sometimes just in a file in their publication. So it takes a lot of manual work to curate those information, but those information do exist publicly. Yeah.
Thank you so much. And what features are available to compare data sets?
So in Synaposis, we offer two kind of cross datasets comparison. One is that you can click up to 5 different datasets and compare them individually. In that setting, because we haven't pre-integrated them, you can compare like something like cell type percentage, those kind of information between data sets. But for our core data sets part, because we pre-selected data sets and pre-compiled them into one mega data set, you can compare. So we already did all the work, let's say batch effect removal, those kind of integration. So you can compare them for differentially expressed genes between, let's say, different metadata groups.
Thank you so much. We're nearly out of time, so I'm just going to answer a couple more questions. But if any more questions are wanted from the audience, please do put them in the chat now. But one of the questions said, what data input format do you take?
Is that for cell ID?
I would say
like.
No, I think it's for you.
Yeah. For our database, you can upload both the sparse matrix format or the full matrix format to analyze your data. Yeah.
Thank you so much. And one final question is about, can you use data sets from species other than human or mouse?
Because we tailored our database more towards the clinical and translational research, currently we only include human and mouse, and we will add the data from monkey in the last few months.
Thank you very much. So that is all that we have time for today. I want to say a huge thank you both Antonio and G for those fascinating presentations, and to the audience for joining us live and for asking your questions. I just want to remind everybody that this webinar is available on demand. As soon as we finish this, you can access the recording at any time using the link that you received in your email inbox to join today. Have a lovely afternoon wherever you are, and we look forward to seeing you at the next one. See you soon. Bye-bye.
Thank you. Bye-bye.
Bye.